Welcome to BioIT World's Trends from the Trenches podcast, your insider's look at the science, technology, and executive trends driving the life sciences. I'm Allison Proffitt, editor of BioIT World. This episode is hosted by Stan Gloss, founder of BioTeam, a life sciences IT consulting company at the intersection of science, data, and technology. Today, Stan talks with Kim Branson, Senior Vice President and Global Head of AI and ML at GlaxoSmithKline. Together, they cover AI ethics, digital twins, and how GSK seeks to build multidisciplinary teams. Let's listen in.
Kim, I'm really excited to have you on our show today. It's funny because I've done a couple of interviews with Mike Montello and also Mason Victors. And the thing that was common between both of those interviews was your name. It came up in both conversations. That's when I knew I had to interview you, because if they're talking about you, then I need to talk to you. So it's a pleasure to have you today joining us on the podcast. And maybe we can just start off. Maybe you could tell me a little bit about your background and how AI and ML works into it.
Sure. It's great to be here, Stan. And yes, both Mike, who I work closely with, and Mason, they're great people. So you've obviously been talking to the right people. Yeah. So look, I mean, my background, I got into machine learning through really being fascinated by drug discovery. So I have a background, I guess, in medicine and science, and it was really thinking. I sort of fell in love with structural biology. And like many people, I read the $1 Billion Molecule book about Vertex. And I sat there and the calculation, I was like, well, if I could just discover one drug, right, that would have just a massive impact on medicine. So I got really fascinated by structural biology, so X-ray crystallography and NMR. And, to me, there was nothing more interesting than like a protein ligand crystal structure. And so I ended up doing a PhD in machine learning, but applying all the machine learning stuff to small molecule drug discovery. So my PhD is really on optimal combination of weak learners, and what those things are in this case are these things that we call scoring functions, that are ways of approximating the biophysical interaction of a ligand and a protein. And they can work really well if if they're tuned for the situation. But if you're going to a new situation, a new protein, a new thing, well, how do you know which one to use and how to combine them? So I spent a lot of time working on that. And that's really what got me into machine learning, and that it sort of happens to be at the right time, where we used to joke, the second best way of solving any problem was a neural network, and there were the state of the art was very different to what it is now. But I've sort of been involved in that field ever since, and always around sort of biology, in biology, and particularly drug discovery and medicine. So I was at University of Melbourne, and then at Stanford for a bit, and then spent some time at Vertex Pharmaceuticals, and then sort of back to the Bay Area doing a series of startups and in various domains, from search to sort of differential privacy, machine learning, and medical records, and then disease risk prediction on claims of the longitudinal data. Was it probably a disease X at time T, given your past medical history, and then to Genentech, and then to GlaxoSmithKline, where I am now. So that's sort of been my bag, right? is really about machine learning in medicine, and it's just a fascination with data and how to use that to predict various aspects of health.
So we hear AI and machine learning as a buzzword these days, but this is technology that's been around for an awful long time, hasn't it?
That's right. I mean, it's you've seen it being applied in other disciplines, and really one of the things that happens in machine learning is really like you have to have labeled data in a computable format, right? That's the key thing, and that's why we saw an explosion in other areas, particularly in the early days of the web, because it was very quick. We could harvest large amounts of labeled data sets from the web and other types of things. And in biology, it was always a challenge because it was sort of a small n, high dimensional complex data, noisy measurements.
Right.
And, it was difficult. And But then, what's happened recently is really this explosion of measurement technology has come to biology. So now with the advent of single-cell sequencing, on a single cell, we can look at the genome, we can look at the messenger RNA, the bits that the genome codes for, we can look at the proteomics, we can function, we can image it. Suddenly we have this massive explosion of data in biology, and the complexity that we can measure it has grown. We also have all these different modalities, right? So I can do clinical imaging, I can do blood-based biomarkers, I can do all these different types of things as well that are happening. I can make iPSC stem cells from you as well. All these things has led to sort of this explosion of data being generated in biology. And I also would say a large fraction of it is, these, having, electronic medical records, but also pairing that with genetics. And that's also been a huge key driver in the adoption of machine learning into the pharma industry as well.
So AI and machine learning, it's been heralded as a way for pharma to bring drugs to market faster. Is that a pipe dream, or is that something that will actually come to reality?
Yes. So the key thing, AI is transforming the pharmaceutical industry, and we've seen it transform and change other industries, and our industry is no exception to that. Where it will transform, it really depends on where you're focusing on the right problem. So at GSK, our machine learning is absolutely core to our strategy as a company. And our strategy is really about sort of, combining human genetics, right, machine learning, and then these sort of functional genomics technologies on the other side. And I'll go what I mean. Why is that so important to us? So we've done sort of studies showing that if you look at known drugs, and now we have a whole lot of different genetic data, we can kind of go back and say, well, was there any kind of genetic evidence that the target of this known drug, the gene product that it interacts with, was supported by genetic data? And if there is, right, those targets that have genetic data, they're about twice as likely to become successful drugs on the market. And so what's happening now is that we have the ability to look at genetics, and now we have all these sort of functional genomics technologies. So these are CRISPR and things like the like. So now I can look and edit and turn up or down the expression of a particular gene in the cell. That allows us to sort of mimic a pharmacological agent. I don't have to go to all the effort of making an antibody or a small molecule that's selective against it. I can turn it up and down through gene editing. And that allows me to then generate a lot of data about that. That particular target, right? To work out, well, is this something we should make a medicine against, right? So what happens where machine learning sits in the middle is now we can generate so much data about those particular targets, right? I mentioned before about single cell sequencing and imaging and all those multimodalities. You throw such a large amount of information plus all the other sort of data sets together that you sort of need machine learning to make sense of it all. So we're at a point now in our company where 70% of our drug targets they're working on have genetics behind it. And you might think, well, that's great. But some of the areas we work on, we're applying machine learning into this early discovery phase. So if you look at large genetic databases that have, where we do a genetic wide association study, and we can come up, well, here's a list of variants that are highly enriched for in the people that get this disease versus people that don't. And then the next question, well, what variants, what do those genes mean? And what it means is we only really know usually what to do with about sort of 15% of those variants we can kind of confidently assign to a particular gene. But our group has been spending a lot of effort in building machine learning models to sort of help with that mapping problem. Right, so we can assign more and more of those variants to a gene with high confidence, right? Now, there are some simple models, like nearest gene would get you there 60% of the time, but that means 40% of the time you don't know what to do with it. And so that's one of the things we're working on is to sort of basically, we're applying machine learning genetics in that sense. And, so basically help us find more targets, help us work out which are great targets to take forward. And then on the other side, actually in the clinic, so towards the clinical end of things where an example of that is sort of our computational pathology work, right? Where, we have, you've all seen those sort of slides, for example, of, there's red-blue slides of hematoxin, eosin stain things. And for there, we can have a pathologist, look at a slide and score various types of things, but we also can build, apply computer vision algorithms to that to do things like quantify the expression of various markers that are important for a drug response, but also then say, actually, not only looking at the slide and saying X percentage of it seems to have high expression, but which cell types and where are they in the tumor, for example, and things like that, and even more detailed measurements. So that's the other part. And then there are areas, such as doing better protein engineering and optimization or small molecule design, using generative methods and things like that. And all those things are putting better targets into the pipeline, being more efficient at moving them through. And even at the end, like the reason why we do these computational biology things are really about can we build models of who's likely to respond, right, to this particular medicine, right? And what's their response going to be? And there it's inherently multimodal. Right, I can build models that take into images of a pathology slide, but also the gene expression of that tumor. Right, so that's that sort of pictures plus numbers, sort of together at last kind of thing. That these neural network methods are really great at combining those. So we see it sort of across the whole chain, but for us, it's really about focusing on those early targets and finding out those genes that are causally related to the pathogenesis of disease. I'm talking that these changes tend to lead to the disease process rather than gene changes in the process we see because of the disease. What I would say is downstream of that, for example.
Right. So if I remember correctly, reading that GSK has a relationship with 23andMe to get data from them as well. So is that helpful in the research that you're doing?
Yeah, the 23andMe data is that's mostly operated between our human genetics department and 23andMe, but that's where sort of targets come up with. So that's a data source where they, we work with them, and we've had a lot of really great targets that have come out of that collaboration. Including one of them, I think, is in phase 1 now, and it's really we sort of help build models that our genetics group can help to apply to work out how to sort of use that data from 23andMe and others. But 23andMe has the largest data set, and this is where the old saw about quantity having a quality all of its own is key, because when the way GWAS currently works is the greater your sample size, the more patients you have, even if you've got some loose phenotype definitions, you can find very amazing things. And maybe one example of that is that 23andMe did if you have a 23andMe, you may have got a question saying, are you a morning person or are you a night person? You think, well, that's kind of pretty vague, and maybe it depends on whether I've got young children or not, or what age I am. Is anyone a morning person or night person? But you ask that with enough people, and they did that, and lo and behold, the genes involved in the clock genes, circadian rhythm genes, popped out of that analysis study. So it does show that with a huge, large data sets, you can really pinpoint some fundamental things in biology, and the clock genes took that's a Nobel Prize, and lots of lab experiments and just software and everything else, right? So it just showed the things you can do when you have a really large data set.
Right. Well, I'm one of those early morning persons, and this may actually apply when you think about dosing somebody on a medication. So it sounds trivial, just do I take this? Am I a morning person or am I an evening person? But you may find a correlation with uptake in certain types of medications that make it more effective for the patient. So even small pieces of data could add up to something very useful.
That's very true, and pharmacogenomics is a very real thing, and there are known associations between particular P450s. These are the enzymes that modify drugs in your body, part of the circulating dose and how you respond and things like that. But also they're fundamentals of intrinsic things, but there are things about the disease, right? About sort of the immune system, and perhaps if you're looking at lupus nephritis, right? It's autoimmune. Right, but I can sequence your germline, and that tells me a bit about you and your predisposition. But it doesn't tell me about your immune system. It tells me about your immune system is what I would call the state function of the individual. So it's basically it's a living thing. It's shaped by your environment and everything else, and the way you live your life and things like that. So we need to measure things about that as well, and that's where also machine learning comes in, because we can profile all those. Different subtypes of T cells and things like that, and your TCR sequencing and your B cell receptors, all those different types of things, and that that's a high complex multidimensional data that we can start to blend together with the genetics data to start to understand, like, are there subtypes of the disease and things like that. And that's another area where we're doing what I would call sort of more clinic in the loop data, as we start to learn from these observational cohorts about disease as well.
So, yeah, I'm sure there are many different subtypes of many different diseases. So maybe I just want to, what are the choices about where you can apply AI in pharma and your take on the industry approach overall? Can you give us a kind of generalized view about that?
Yeah, I think that you have to be very thoughtful about where you apply your machine learning, and it needs to be areas that have a large 1000x impact. And I think that you don't want to do like a tweak to something that you make a 5% change to a process probably isn't meaningful, but allowing you to do something else at a scale or speed or something you couldn't do before, right, is really important. So I think choosing the problem is very, very is key for us. And we think that choosing the right problem will give us sort of a step change in our innovation. So picking the right targets, right? Like a lot of things fail just because, well, that target, it was correlative and things like that. But when we made a medicine against it, it just didn't move the needle in the clinic, right? So we know there's a lot of different measurements, right? It's very clear that, blood-based biomarkers won't do it alone. For oncology, I need to do imaging. I need to do other types of things, right? So bring all that information together, right, to characterize the disease, but also to come up with the models that we study things in, right? Like the various experimental systems and how well they link to the patient is very key. So we're doing a lot of work on, we've talked about discovering targets, but also basically building, and this is borrowing terms from engineering, bridging models, right? So
right.
A lot of people are like, well, I've got this animal model of X, and will say, well, okay, it's a model. What's its positive predictive value? What bits of the clinical disease does it predict? And then they look at you blankly, like, well, what positive predictive? What are you talking about, Kim? And then you realize, like, okay, well, we have to say, like, I'm a machine learning person. I look at things very different. I'm like, here's the disease. Well, what elements of the disease does it capture, right? So what's its representative capacity of the disease, and what's its fidelity? How close is it? And what bits does it get there? And you realize that you need to actually start to look at populations of people and look at that sort of stuff and build a bridging model, right? That there's a clinical translation piece. Because if I'm studying a whole bunch of things in my cellular system, which is cheap and easy to manipulate, but it has no bearing to what happens in the clinical progression, and I'm not. It's the ground truth is all wrong, and I can do all the machine learning I want and all the active learning within that cellular system, but it'll be all wrong. So I have to understand. And build a model of that translational aspect. So we're spending a lot of work on that. We've got a whole new institute for doing that with this Oxford Centre for Computational Molecular Medicine that GSK set up, where we're building these observational cohorts and building these types of things. And, we can measure things in people, we can examine lots of sort of stuff, but we can't sort of, we have to have ways of bringing that back into the lab in a system that we can sort of perturb. I can do my CRISPR edits, I can learn things, but it has to be a meaningful system. So we're building models for that as a very key thing, right? And that's what I would call sort of that data in itself is inherently multi-scale, multimodal, because you can imagine if I have a cellular system that's a single cell, well, it's not going to capture multi-organ disease, right? In human, right? But maybe it's a really good model of a fundamental cell in the liver. So maybe that's mostly what I care about. So it's good for that, and I can work out how to bridge it. So we do better early research as we start to sort of bring the patient into the loop, right? And really engage those patient populations. So that's a key area.
So, Kim, I really appreciate that last response in terms of thoughtfulness. That was one of the first words you used, to be thoughtful in the choice of where you use AI and machine learning. It's not necessarily needed everywhere, but doing it targeted in the right place. I think that point is really, really important. So I would love to understand how GSK applies AI and machine learning across the continuum from R&D to clinical trials, because I think we've touched upon it, but kind of where are the touch points and where is value derived of using AI and machine learning across the continuum?
GSK takes a very different approach to most pharma companies with the way we've integrated and used AI. And so the first thing is that we, as an AI group, work on only a few problems, right? So we put a lot of our effort behind a few different vectors. And the first one is on those target discovery, right? So applying machine learning to the sort of genetics and genomics and those aspects to come up with targets that are causally related to a disease. But in the process of doing that, it does all come down to the data you have to learn from. And so for all of the AI systems we build, uniquely at GSK, we have sort of lab in the loop, right? So we actually have a machine learning algorithm that we're doing experiments for, and these are automated robotized experiments, things like that, to generate data specifically for the purposes of building that algorithm. So typically you might come to a company and they're like, well, we've got some data that's relevant to what you want, but that data was sort of generated under a biasing constraint. So it had it was for another purpose, right? But I want to build a general purpose tool. And so what we actually do is we generate data for the model to build a general purpose model, but we take it one step further. We actually ask the model what data is most informative for it, right? So rather than saying, well, here's 1,000 data points, and I might look at it and there were like 700 of one class and 300 of another class, and I've looked at the first 200 of that 700 class. I'm like, I'm really good at this. I didn't need the rest of it, but I've never really cracked those other 300. I would have liked more of those data points. We sort of ask the model, like, what are you most uncertain about? What things do you want to have to know? And we generate more of that. So we turn everything into what we call a sequential learning problem. Mm-hmm. And so we've done a lot of work of working with our colleagues in comp bio and functional genomics to build these systems, right? But biology is hard, right? It's not like other parts of machine learning. Like cells take time to grow, reagents take time, but you can compress that and bring that down and automate it. So we can basically run this loop where we're basically continuously generating data. And so just sort of as a point on that, I mean, last year we generated another 21 billion data points in sort of primary human immune cells for helping us work out this sort of variant to gene mapping, right? And so it's that body of data we're generating, particularly for machine learning, that becomes unique for us. And so then that generates this tool that our colleagues in human genetics and comp bio and other groups can really use in their drug discovery. So that they can map more variants to more genes or things they couldn't do before. It gives them different things to look at. I mean, another example is in the area of oncology. So there's something called a concept called synthetic lethality. And that sounds very, very complex, but it's really quite simple. I mean, in biology, like most things, right, we have redundant pathways, right? There's two copies of a way of doing something, right? Of like, because you don't want to, organisms only had one way of doing something. It doesn't take much of an insult, and the whole thing sort of falls over. So as tumor cells kind of grow, and tumors are clonal disease, as they grow and things like that, they're dysregulated with their growth. They tend to get mutations in like something called a pathway. So they might lose one copy of an important bit, but they've still got the other bits. They're fine. If you can determine which one of those other things that they typically tend to lose, and you make a drug against that particular part of the pathway, and that pathway is involved in their viability, you take it out, they fall over, then you actually get something called its selective toxicity, right? So that's a synthetically lethal. Synthetic means it's lethal because it stops the cell growing. Synthetic means it's a drug that comes in. And that's the core about what, one of our drugs, niraparib, right? But it's a class of that kind of drug. And we have been, obviously, there's one class of that. Well, there must be other ways there must be other particularly synthetic lethal targets. So we, built a whole AI system for sort of coming up with that sort of information, where we are looking at, a particularly, what I'll call side A, a particular gene that gets lost in tumors. And we want to then say, well, if I inhibit this other pathway, does it stop the whole thing? Does it kill the cell, or does it kill the cell? I don't care about that. I didn't need that anyway. And to come up with other synthetic lethal targets. So we've been running a machine learning loop to build a particular model for this. And this work is coming out of our team out of Tel Aviv there. And then we found 8 targets that everyone's like, oh, wow, I would never have thought of that. There's no literature. It seems to work. wow, let's go and like really look into this, right? And so, there are efficiency gains. There are things coming up with new targets, right, that are coming through as well. And then there's also, as we go towards the clinic, we have AI people involved in the early pre-phase 1 stuff of some of our assets, but also in the area of oncology, for example, where we have 4 different great IO drugs, and IO is one of those amazing advances in chemotherapy where 20% of the patients can get miraculous, they're cured, right? And you're like, wow, that's amazing. Which 20%? Oh, we don't know. Right. And you're like, well, that's how we should definitely work on that. And so what you don't know is like, well, these immune checkpoint inhibitors, do we just add more of them, different types, and what tumor types should you give them and when and things like that. And so we're working on that aspect because that's really foundational, because we can't just do the combinatoric of all possible IO agents we have in our toolbox in all different tumor types, right? It's too expensive. There's too many, right? If we just have 4, there's 64, combinations of alone. If you think of which order I give them in. So we're starting to do things where, and this is with our work with King's College London and Professor Tony Ng, where we're making these biological twins of tumors. So we will literally take a patient's tumor that's been resected, we grow their tumor up as something called an organoid, or it just sort of means it's like a 3D tumor culture. But then usually we take their immune system and their relevant cells of the immune system and we add it to that tumor, and we see how their immune system responds to it. And then we can add various combinations of those immune drugs. And we're trying to work out, we have sort of a model of their tumor in a dish. And we're trying to work out really, working out the rules, right? We're trying to work out what things to give when, what drugs to give when from this. And this is all our sort of idea of like, well, we're kind of trying to play chess with cancer, right? I want to treat you with this drug, right? And I don't want to get resistance, but I know if I treat with this sort of thing, then you're going to have clones that are going to appear that are susceptible to this. And I have another thing I can come in with, right? So we can kind of like play a longer term strategy rather than just I'll treat with this until I see resistance, and then I'll see what I've got next to do. And thinking how to even personalize that based on the characteristics of their immune system and the tumor they have. So that's another area where we're pushing on things that's sort of closer to the clinic. And then there's areas of really, one of the first places to start is computational pathology, where we are we're building models, and one of the key things to understand for IO response is that where is the target expressed? Does this person's particular immune system, their tumor, express a high amount of this target or not? And, we have ways of doing that. We have an antibody against that particular target, and we can stain the pathology slide and we can look for that. And it takes a human to go through and score and that sort of thing. And then they typically characterize on a fairly broad basis, right? Because they look at the slide, they all the X% is that. And you would give it to 1 or 2 pathologists and they look at that and you might average the result and things. But what we can do with AI is like, well, this is really just picking brown pixels on a slide. I can build a machine to do that. Mm-hmm. I can build a machine to quantify that rather than yes, no, a continuous amount. I also can build a machine that can label which cell type it is. And we have a concept in IO, people will talk about immune deserts, and all they mean is like, are there immune cells in the tumor or not, right? And things like that. But you can start to measure the different types of cells, where they are on the tumor, whether they're expressing that or not, all these types of characteristics, a lot more things that we could do before that a human can't do. So it reduces the variance of the sort of aspect, but it allows us to measure a whole bunch of other things. We're starting to take those into the clinic. And when we get the response data that's paired with that, we can now start to build a model based on the pathology of the response, right? So that's like another area. So we've just done we've seen in the press, I deal with our good friends at Path.ai, who are a great computational pathology company. And, I was talking to their CEO and I said, well, I've got some great models. I really want to get these to the clinic. He said, well, we have a fantastic platform for doing that. I mean, we build models for other people, but you could just work with us to put your models on our platform. I'm like, this is going to be fantastic. So that's the sorts of things where we're trying to sort of push into the clinic now. But it does all come down to, to do AI successfully at pharma, there's a cultural piece, right? There's the data piece. There's thinking about iteration cycles, right? So, Like we think in terms of generate data, generate a bit of data, make a model, generate it again. And when we started, we that's part of the whole cultural transformation that we've been been working with, right? And it's been really fun to see the biology that's like, okay, 2 years ago I didn't get you guys, now I get it, right? And vice versa, right?
So I just love that whole answer that you had. Just one of the things that jumped out at me that we could probably do a whole podcast on is this concept of digital twins. It's just a mind-blowing concept when I stop and think about it. But I want to pivot over to a different topic. And not too long ago, I was sitting home watching a 60 Minutes episode on AI and the challenges with African-American people getting biased by the AI algorithms. So let me ask you, what's your advice of how to reduce and eliminate bias in your AI analysis? And kind of what are the ethical issues here, especially, in clinical trials where we saw in the pandemic, we needed a diverse population of people to make sure the vaccines were right?
This is a hugely important question, Stan, and something that we've been thinking about, ever since we started the group at GSK. The ethics of this is front and center for our mind, right? At GSK, we make medicines for everybody, right? Like that's our goal. We make medicines for everybody. We don't make medicines for a subgroup or anything else. It's for everybody. And the same thing, we want to make AI approaches we do that need to apply to everybody as well. And we looked at this and we realized that, so I've talked a bit about feedback loops of going into things, but the entire drug discovery process itself is a feedback loop, because the data that we're mining for our genetic databases and the medical records and things like that feeds into the system. And we run the GWAS, we run various types of things, and we see what we can study and find out. And the targets we have, right? It depends on the representation in that, and it gets amplified all the way through. And then we started thinking about like, okay, say I've built this amazing new way of predicting response. It just needs your full gene sequence, the sequence of your tumor, immunophenotyping, and an MRI of your tumor, right? Okay, you can get all that in North America. And maybe there's some way we can combine all together. But thinking about developing countries, well, the data infrastructure to do all that and pull it all together in one place and then apply the algorithm. Well, where is the data infrastructure? And we suddenly realized we need the equivalent of almost like a data cold chain, like we have for vaccines. For vaccines, you have a cold chain of how do I move the vaccine around? Everyone now is familiar with cold chains and vaccines, right? So we have, and the other thing we care about is like the robustness and reliability. If we put these things on, they have to work, right? Like making big decisions, right? So we created a position, and we have a fantastic guy, Robert Vandersloot, who's our VP of AI ethics and policy. And as part of that, we realized that we looked at the ethics community and right, we want it to be part of what we do, and we want to think about how to change the system, and that means inclusiveness in trials, and how do we do sensors and wearables to make sightless trials and things like that. But also, there are big questions about what we're working with, right? That there are ethical issues that need to be investigated. So with Stanford, we've set up this new program to recruit engineers and people that to come in, and we teach them ethics, or people in other disciplines to teach them bioethics, but to do research into these types of questions, right? Because really good public policy needs to be formulated by a realistic thing of fears, right? So I don't know if you remember the early days of nanotechnology where everyone was like, oh, grey goo scenarios and things like that. And even working on it was like, that's laughable. We're so far from doing that. I can barely move a carbon atom over here, sort of thing, right? There's this same thing with the AI community. There are some groups that are coming from outside, and they don't have the technological training, so they don't know how to assess type stuff. And some of the things they're talking about are very hypothetical, and some of them are real, right? And a lot of the machine learning people can kind of sweep some of the sort of stuff under the rug. But we needed to kind of create this new kind of like let's investigate real informative things now, so we can write design policy and we can make good policy suggestions on how to do things. Because we fundamentally believe this is a transformative technique for health, and we don't want to miss the opportunity. So we've created this group. There's a series of ethics fellows in Stanford, and this is a global thing, so anyone can come. And Robert's going to be at a conference in West Africa soon. So I'm hoping that we're going to get people from all over the world. And we've got another set of these fellows appearing in Australia as well, the Australian Institute of Machine Learning as well. And so this is a very key thing for us. And so it has to be, yes, how do we actually have a whole thing about when you start a machine learning process. We have a series of checklists in the way we think about things. And if you say, well, I've got this is the data I have, and like I'm missing this group, well, it's not okay just to say, oh, well, I just didn't have it. It's like, well, fine, go get more data. What's the budget? What do you need to do this? Right. Mm-hmm. And it's absolutely key. So, and you'd be like, okay, well, I can buy more data. Like, of course you can. Like, let's try and do our best effort. Now, if it's hard to do it or we can't collect and things like that, and you will get different performance, right? Just the amounts of things. And maybe that reflects the true nature of the disease or not, but you have to try. It's not okay to sort of do it after the fact. We also care about what's the effect of the system when you deploy this, which is that research that I was talking about earlier. Like, mm-hmm. How will this change the healthcare system? So we care very deeply about this. And so for us, it's, GSK is a 300-year-old company, right? And like that's a strong, continuous reputation of trust of many generations of scientists that we're just the current generation of. And we want to kind of sort of continue that agenda, right? So for us, it's really front and center. So I'm very glad you asked this question. It's something that, as you can see, I'm quite passionate about, and we could probably again fork off to another podcast.
So actually, one of the points that you actually touched on about retraining people to have skills to work in the area leads me to kind of wonder, how do you recruit talent for your team? And is that team a multidisciplinary team?
So I would say most of our team in the GSK group, so it's about 120 people, they're brand new to GSK, right? They didn't come here. And so I think a lot of people, what a lot of people didn't realize is that, it requires a leadership, right? So Emma was doing a transformation of GSK. And before that, I was like, I talked to GSK. And then people were like, well, see, Hal's there. And I'm like, oh, Hal's there. Okay, I know Hal. And people, and like, Hal's there and Tony Wood's there. And like, they really get it. And I was like, you talk to them like, okay, you guys really get it. We're serious. And so you have to kind of create the environment to say, well, by the way, AI people are not data scientists. You need data engineering. You need a whole different support stuff. They're the tip of the spear, but there's other groups you need. And they need experimental feedback, and they need data. And they're also, we come and work in a very different way. So, we're sort of decentralized organization. We're spread over different types of areas. I'm in San Francisco. We have people in Tel Aviv. We have people in Heidelberg. You name it. And our core group in London. We work in a different way, but we recruited in people to do this. And really, there's a generation of people that are like, I'm like, look, I've got computation. I've got systems like Cerebus. I've got plenty of GPUs. I've got a really interesting problems, right? And we can generate more data, right? That's really impactful. And there's a lot of people like, actually, and now is the time to do machine learning and bio. There's a lot of people that are interested. It just wasn't at the time. So there's a lot of people like, okay, this is great. I get what you're doing. I want to come here. I want to be part of this. And so I, like people like Jeremy England joined us from MIT, and I've known Jeremy for a long time. So to Jeremy, I'm like, this is the most important stuff we can do. We've got really fantastic data. We can generate more of it. It has a huge impact on human health. We can, there's a lot of people like, well, if I could do things now as an academic, I have to do a startup or something else to commercialize. I'm like, no, you can push it in there, right? And so we've also, got the next generation of fantastic leaders like Katie Aiello and others that leads our variant to gene mapping project as well, that are coming in, that are drawn to this. But this is a really important place. This is a place that gets it. They're supporting us well. And for machine learning people, the most important thing is actually like, we really want to, we want to get our scorecard back. So we built a model, right? We want our score. And so right. That will generate more data for you, and we'll really check. And we want to work with you on this. Like, yeah, maybe the first version was bad. The second version is a little bit better, third better. Now we're getting it. like that cycle is really important to people as well. And also creating the right culture. So people don't want to come and vanish into a black box. So I'm like, no, we're part of the machine learning community. We contribute to open source packages. We publish our code, right? We'll publish our code. We'll publish things at papers. If it's a model on public data that everyone else has, we'll publish our model built on that public data, because why not? Like, you can take it and use it for what you want to do and tell us about it. The models on the private data we generated, things we're using, the model that we're not going to release that. Obviously, that's our job. But we definitely are part of the community, and that's allowed us to attract people. A lot of people are like, I didn't realize that was a thing. And so we've had people come in. We've also created this AI fellowship program. So we have this GSK AI fellowship where we kind of put our calls, like, if you've done machine learning and a PhD in machine learning in a different discipline, for example, I have a great person, Emma Slade, who did her PhD in particle physics, that came into an AI fellowship program, and we teach them a lot about pharma, drug development, bio, and they work with us sort of for 2 years. A lot of them are like, actually, this place is awesome, right? I want to stay. This is really cool. And some people are like, well, this is a really fun experience, but I want to go run an academic lab. Or actually, I'm still exploring. This is a great fun 2 years. I'm going to go off and do this stuff in finance now. And so but it's been a great program to bring those people in. And so for us, we want to have people that come in with different mathematical toolboxes.
Right.
So I come at the world through, machine learning and really an information theoretical kind of approach to that kind of aspect. Jeremy England comes from a statistical mechanics, physics kind of perspective. Kate is coming from bioengineering with that. Emma's coming from particle physics. Everyone has different mathematical tools and ways of looking at a problem. We have people that are pure mathematicians and topologists as well. But that whole idea where we can look at a problem, everyone has different ways of looking at it. Something magical happens with that. So we are very careful to kind of create a culture where everybody is sort of free to kind of work on things and contribute. But we're not just hiring, 15 Kim Branson clones. Because I kind of know what I think, and I don't need 15 other people reinforcing my design. Having that intersection, that's where we have really innovative things happen. So if you create the right environment for that, so that's been a key to us. Absolutely key to our hiring, and that's the philosophy we have. And we also have specialists in sub-disciplines. We have people who've obviously done lots of clinical imaging and machine learning and clinical imaging, right? Like, you have to obviously understand the domain as well. But having those different tool sets and different mathematical toolboxes and machine learning toolboxes, experiences in different industries is absolutely key.
Well, Kim, thank you so much for this conversation today. I mean, I've circled it on my calendar for months now, waiting for us to have this conversation. And I am so glad that we were able to capture this conversation and share the wealth of knowledge that you have in AI and machine learning with our audience. So I want to thank you very much. And I think we'll be talking again for another episode. I got digital twins on my mind.
Yeah, we'll definitely have to bring in my good friend Tony Ng, and we can definitely love to talk about digital twins. There's so much to talk about there. But this has been a great conversation, Stan, and thank you.
Thank you very much.
This episode was produced by Pete Glowacki and myself, Allison Profit, with the BioIT World media team from www.bioitworld.com. It was hosted by Stan Gloss, founder of BioTeam, a life science IT consulting firm at the intersection of science, data, and technology. BioTeam builds innovative scientific data ecosystems that close the gap between what scientists want to do with their data and what they can do. Learn more at www.bioteam.net.